Good afternoon and a very warm welcome to the second in our series of seminars on technology and the future. We are extremely fortunate this evening to have two treats in store for us. So it is actually someone who was just outside their birthday, and this is the birthday treat. Two real experts on different dimensions of nanotechnology. So if you don't know anything about nanotechnology, this is an incredible introduction. If you know a lot, I would imagine these people know even more than you. So there'll be lots of questions. They've been working on it, particularly Eric has, for longer than virtually anyone in the world, and was one of the pioneers of nano, was one of the people to really begin to think about it in the 1980s, made the concept of nano more widely known, and subsequently has thought extremely deeply about many dimensions of it, not least the societal, broad societal, the manufacturing, the business, and other implications of it. We're extremely fortunate in the Oxford Martin School that we've been able to attract Eric here to work with us. He's been here for 5 years now. He's just told me he has no intention to leave, so that's a treat for us all as well. I'm delighted to hear that because he finds this a great environment to work in, because we work on the big challenges of the future. So Eric is a senior visiting fellow here. Maybe we'll have to remove his title visiting after 5 years. He's a senior fellow here and a pioneering nanotechnology researcher and author. His 1981 paper in the Proceedings of the National Academy of Sciences established the fundamental principles of molecular engineering and identified many of the ways that it would evolve in the future. His 86 book, Engines of Creation, introduced nano to a very wide audience, and he's continued to take this forward, and his latest book, Radical Abundance, has gone even further in thinking about the implications for society of nanotechnology. So in many ways, I think it is fair to call Eric the father of nanotechnology. He'll be joined, following his presentation, by Sonia Trigueros, who's an Oxford Martin Senior Fellow, an academic fellow in the University of Oxford's Physics Department, and co-director of our program on nanotechnology. And Sonia has been working with us for a long time on developing specific medical applications of nanotechnology, and is an absolute leader in her field in thinking about how nano may be applied in many different areas. And every time I hear her, she's come up with a new possible application of nano, from cures for cancer to antibiotic resistance to many, many, many others. So I'm always delighted to hear her. After her postdoctoral fellowships at Harvard and Oxford, she's gone on to be a visiting fellow in many places, and we were delighted that we were able to attract her and retain her here to be the co-director of her program on nanotechnology. So I'm delighted they've both agreed to speak in this series of lectures on technology for tomorrow and the research agenda which is shaping our future, because this clearly is absolutely that. Eric, the floor is yours.
Thank you, Ian, for that wonderful introduction, and for the opportunity to speak to you all this evening, and for the opportunity to be at the Oxford Martin School and to have an Oxford Martin School in existence to be at. Great, thanks. So, this afternoon, I would like to outline to you the prospects for a turning point in the development of nanotechnology. In the early days of the concept, the focus was on a long-term objective: flexible, programmable, atomically precise fabrication of intricate objects, including components for machines that could do a better job of assembly of intricate atomically precise structures. During that time, we have seen the slow maturation of the development of ways of making molecular structures that are adequate to get us to the first rung of the ladder. And in the last few years, a set of technologies has reached a threshold that will enable us to take a crucial step, and I would like to describe that step to you today. This presentation, this topic was first introduced last August and has been developing some momentum as a research goal since then. So the large transition ahead is from molecular self-assembly to molecular additive manufacturing. And I'll be hitting several topics here. One is where macro-scale fabrication is today, the way we make things in the macroscopic world by systematic engineering. Atomically precise fabrication today, which is very different, very contrasting. Then how to build on that to develop a first generation of atomically precise molecular additive manufacturing systems. And then just a few words about early applications, which I will turn over to Sonia very promptly because she is the one who is working in the area where the greatest applications are likely to emerge. So first, macro-scale fabrication. If you look around you, you'll see many artifacts. You'll see people and who are not artifacts, but virtually everything else you see was made by machines or with the aid of machines. And those machines were made by machines or with the aid of machines. And tracing back the genealogy of machines, you'll find earlier and less automated and simpler machines. If you go back far enough down the family tree of industrial technologies, you'll find someone doing this kind of work. Shaping materials in a direct way to make components and tools that could make better tools, more mechanized, more precise, that were used to make better and more complex tools, to make better and more complex products, and ultimately a very intricate technology base. Making complex objects has required complex machinery. Now, we'd like to be able to make complex objects in the molecular world, to start climbing a ladder of technologies like the one from blacksmiths forward. But it would be very nice to not have to build very complex systems to make complex objects. And in fact, in recent macro-scale industrial progress, we've seen a way of doing that. You have relatively simple machines, do X, Y, Z motion of something with respect to a platform on which one is building something. And the something that is moved takes some action that results in a little bit of material being added. Sometimes it's using laser to sinter a bit of powdered metal. Other times it's a nozzle that's putting down little bits of melted plastic, the most common form of 3D printer or additive manufacturing system. And the reason this is so attractive is that rather than having lathes which cut cylindrically symmetric objects or various other mechanisms for casting with special tools and so on, where many different machines are needed to make different shapes, instead additive manufacturing, by adding small amounts of material at a time with X, Y, Z control, a bit of material here, a bit more there, can make a tremendous range of shapes, including today aerospace quality materials that are used. This is a component that Airbus is putting in aircraft that you may fly in, and was made by a 3D manufacturing technology, an additive manufacturing technology. Well, the essence of such systems, again, is some structural framework. Moving an X, Y, Z requires a structure to hold parts together, to hold a tool with respect to a workpiece, and stepper motors to increment on X and increment on Y and increment on Z to put the tool where it needs to be to add the next bit of material. So we have structural frameworks and stepper motors, and then some way of getting, in the typical case, materials to the location and then placing those bits of material. Not all 3D printers work that way, and what I will describe in the molecular world avoids that bit of complexity. So atomically precise fabrication today, back up and ask, how do we make complex things from molecules today? Well, the complex structures that are readily accessible today using chemistry, but also biology, because they're biomolecules first and foremost. One is DNA, which forms rod-like objects. The helix, the double helixes, can be thought of as a funny kind of twisted rod that can be used as a structural member. The other class of biopolymers that we can use to make structural objects and functional objects are proteins. My first paper in this area in the Proceedings of the US National Academy of Sciences was on the prospect of protein engineering. People thought it would be impossible because it was too complex to predict how a chain of amino acids would fold into a three-dimensional object. I pointed out that they were trying to predict how natural proteins would fold, and it's a very different task to design one that would fold the way you planned. And people said, aha, and that's the root of the citation tree. In this area where people have gone off and done brilliant work that is now coming to fruition in the direction that I had been looking for as a way of implementing the kind of system that I'm finally in a position to implement after all these years. So it's rather an exciting time. So the nice thing about proteins is that they're highly functional materials. They can be as solid as wood. They have shapes that can be tailored in great detail. They can serve as components for motors. They can serve as components for enzymes that serve as enzymes that transform other molecules and so on. So DNA is simple and soft. Proteins are a very good engineering material. The great advantage of DNA is, in fact, that it's simple. There is a field of technology known as structural DNA nanotechnology, which can now make 100 nanometer scale objects on a scale of, in terms of material content, millions of atoms. And if you follow design rules, making a new object is about as much of a research project as carpentry. There are ways of holding the helices together in a systematic way that all is based on Watson-Crick base pairing. And so these objects can be designed and made in a matter of days. Proteins have recently reached a threshold where engineering self-assembling protein objects, as of 2014, this report, reached the point of being able to systematically design complex objects. Here we have extended arrays, flat sheets, you can think of those as plates that could be used for structural purposes, work done in 2015. And the way this works is that you have to synthesize molecular chains using chemistry or biology. First, designing molecules that will self-assemble in solution. They have to have complementary surfaces. DNA, that's very simple, Watson-Crick base pairing. Proteins, it's a matter of designing pieces that will fold in a predictable way to make what amount to 3D puzzle pieces that will then fit together with other pieces to make a larger object. That's difficult, requires computational search. That's been a long process to get to the threshold that was indicated by the previous two slides. So this is very different from macro-scale fabrication. You don't put the pieces where you want them to be. You do a very complex process that enables self-assembly, starting with one-dimensional chains. So what we'd like to be able to do is additive manufacturing, where you put the pieces where you want them to be. You can see that would be a fundamentally different way of manufacturing atomically precise structures on the nanoscale, enable faster, more systematic design, a wider range of materials, be a technology platform for a fundamentally new way of making intricate nanoscale objects with atomic precision. That is a very large prospect, and now finally there is a clear picture of how to get there. 3D printers require structural frameworks and steppers. It's still necessary. As for the tip functionality, want to simplify systems, you can substitute bulk fluid flow and diffusion to transport materials, and instead of putting pieces where you want them to be, you can take a workpiece and activate a location so that the building blocks that are washed in bind there. So three-dimensional designation of where the blocks go rather than moving the block, much simpler than what people had thought just a few years ago. Molecular chains are made to do by chemistry. You start with a block attached to a surface, activate a site chemically, wash in a block that reacts there, and then continue by activating at the end, repeating, and building a chain. And those are the kinds of chains that will then fold up to make objects if you're very clever and design such things. So the concept of 3D additive manufacturing with molecular building blocks is to have a tool, not a solution that washes through and activates the end of a chain, but a tool that can move around on a structure and activate a particular location. So moving to a particular place, activating the site, moving along to additional ones where you'd like to have the same next kind of block, wash-in blocks of that kind, and then iterate additional cycles, additional layers, different kinds of blocks on each cycle to make an intricate three-dimensional object. This is a stack of bricks. There's no reason why the object has to be that simple in structure. So what's required for that are the machine and the building blocks and the tip for activating locations. If you have that, you can avoid folding chains to make puzzle pieces, have greater design freedom, wider range of parts, structures, and functions. Fast design and fabrication for product development, put the pieces where you want them to be. And a key point here is that if the structure, as I'll be describing, is based on self-assembly of DNA and proteins, for example, and some auxiliary molecules made by organic synthesis, you don't talk about how many devices you're making. You talk about how much material is being self-assembled to make those devices. And in chemistry, a milligram is considered to be a respectable but small amount of material, and a gram is considered to be modest but very substantial amount of material. Machines of this sort would be on a scale of 100 nanometers or so, and a milligram of material being macromolecules assembling to make those machines provides 10 to the 12th at a shot. 1 gram, 10 to the 15th. So rather than having some large machine that's trying to do precise manipulation on a surface, instead the picture is of having a substantial volume, fluid flowing through carrying materials, 10 to the 12th to 10 to the 15th machines working in parallel to make things, to determine how those building blocks are arranged. Hence, scalable quantities, and eventually, by building arrays of machines, there's a pathway to scalable product size. Initial products would be on the scale of the machines, however, which is 100 nanometers or so. So the key components need building blocks, platforms to build on, activation tools. Those are questions of chemistry. There are many ways of solving those problems. None of them are obvious. Chemists do use very clever and indirect methods to make structures. What's needed here is clearly within the envelope of what chemists have done, but we're going to need a bunch of chemists to actually figure out what blocks they can synthesize that meet other system constraints, what the functional groups are that are activated by what kind of catalytic mechanism, and so on. The part that has been the sticking point, the hard part, has been the machine. And there, the two parts, again, are structural frameworks and steppers. So we need a system that can move in X, Y, and Z. X and Y can be performed by strips of self-assembled structure that are in a framework something like this. Z-axis by moving the two U-shaped pieces with respect to one another, and each one is carrying the X slider and the other one the Y slider, and that provides the X, Y, Z motion. The reason that this is a boxy configuration is for structural rigidity. Biopolymers are relatively soft, relatively low modulus. All else equal, lower modulus means larger thermal fluctuations. Thermal fluctuations in a linear elastic model give a Gaussian probability density distribution for the position of the tip. And the thermal fluctuations are constrained by mechanical forces. The variance is inversely proportional to the mechanical stiffness. And if you have soft materials, you try to build a structure that is as rigid as possible with those materials, and one that I just outlined turns out to be a very, very attractive design in that respect. And then the next way of accommodating thermal fluctuations is to have the size of the blocks and the targets be far enough that the probability of hitting the wrong target is very low. So numerically, those constraints can be met in a part of design space that results in some very attractive systems. The other part, which is more interesting, and really some new ideas here, structural mechanisms who are waiting for the fabrication techniques. What's new is a way of making stepper motors that seems very attractive. Probably other ways of doing it, but this one is nice. In the macroscopic world, there are what are called three-phase motors, stepper motors in this case, where turning on one set of windings produces a magnetic field that produces a potential energy function that aligns a moving part with respect to that. Turning that off while turning another one on shifts the location that the moving part wants to sit. You turn that one off, turn on another, it keeps shifting. You do the 3 in reverse order, moves the other direction. So each cycle moves a step. Because molecular actuators are at a premium, there's a trick where you leave out one of those steps and have it be kind of an idle step, and that gives 3 distinct states, which is enough to move left and right in increments. How to do that? Well, want to have 3 input channels for control. Well, a natural way to do control is by flooding the volume with light. If you can have absorption at 3 distinct wavelengths, you have 3 different addressing channels for your actuators. Turns out there are some dye molecules that absorb very nicely with well-separated peaks, modest amount of overlap. And by putting in light at 3 different wavelengths, you can activate different sets of molecules that are activating different phases of a stepper motor. What does that activation look like? Well, there are molecules of a family called azobenzenes, which when they absorb a photon, they flip around a double bond from a trans configuration on the left to a cis configuration on the right. That's enough to change the way they affect their environment, and if that changes the potential by kT, which is easy, that's good enough. Now, there were 2 problems when I was first thinking about this a few years ago. One is that these absorbed light only at very short wavelengths, the violet range. You couldn't channel multiple wavelengths of energy in there because you can only run the energy downhill to longer wavelength absorption. Well, in 2013, developed some clever people who were trying to get systems that would work in the body where red light travels more easily, figured out how to make molecules of this kind that switch with red light. The other problem was that the reverse switching, what took minutes to hours, that time has now been reduced to the microsecond or nanosecond range. Very recent developments, 2013, 2012. So we have the structures, due to recent advances in protein engineering and structural DNA nanotechnology. We now have components that are suitable for motors. So those are the key components. The key challenge is coordinated cross-disciplinary development. Almost all of this work, in fact, actually each of those technologies has been driven primarily by an interest in biological applications, biomedical applications. Researchers who are developing molecules for introduction into biology do not work together to build complex systems built from those molecules. It requires a systems engineering approach where there's a top-level function. You decompose the function into component elements, such as a framework versus a motor versus a control system, chemistry for building blocks. Those need to be put together. And last August, there was a meeting in Berkeley, meeting on integrated nanosystems for atomically precise manufacturing, which is exactly the direction that I've been pointing for some time. Person who this was held by the Advanced Manufacturing Office of the U.S. Department of Energy. The program manager is someone who I first propagandized when he was a doctoral student at MIT back in the 1980s. So another element that has come together is an organization that has a reputation for doing systems engineering. I guess it grew out of the Manhattan Project in part, and they also do renewable energy and many more peaceful developments. And they're now very seriously looking at developing this class of system. They are, however, moving at government speed. The workshop was in August. Apparently, the final workshop draft, well, they've assembled the material for comment by the workshop participants. I'm expecting to see it any day now, and sometime after that, there will be a report from the workshop. I can report that there is also some preliminary interest around the Oxford community in addition or instead having a center of coordination for a systems development effort in this area in the Oxford milieu. We'll see where that goes, some very, very preliminary thinking there. So early applications, well, discussed a lot of them at the workshop for DO8, many of them involving materials processing and strong materials and filtration membranes and electrocatalytic membranes for fuel cells, energy-related applications. But the most attractive applications, I think, are in diagnostic and therapeutic products in medicine. Our bodies are full of atomically precise structures, nanoscale structures, these structures interact with one another, which is how biology basically works, and to be able to make structures of that kind more rapidly, with more design flexibility, more control, and straightforward control of three-dimensional structure can be very important. To give some sense of the complexity of structures that people are investigating today with more conventional means of assembly, here are some nanoparticles. One class of applications of nanoparticles with tailored molecular components for nanomedicine. But at this point, I would like to turn the presentation over to a researcher in nanomedicine who can tell us what kinds of things are being built today that we could perhaps, by providing better tools, enable to move forward even more rapidly. Thank you.
Thank you, Eric.
Excuse me.
Good afternoon. First, I would like to thank the Oxford Martin School for inviting me to give a talk today. It's an honor for me to share the talk with Eric, as Ian pointed out, the father of the nanotechnology. So for me, it's a great honor to be here. And my talk today, I would like to point it out, what is nanotechnology doing for medicine? So when I start thinking about and you start looking on the literature of how many things are now going on in nanotechnology that will change, or is actually changing medicine, I try to group in 4 different advances, like to make it simple. So one is in nanosensors and diagnosis, as Eric already have pointed out. There is a lot of advance going on the nano drug delivery systems, nanotechnology in regenerative medicine, and a lot of things going on nano devices for medical surgery. The nanosensors and diagnosis, what's the philosophy at the nano level that we have on this? So I'm sure that you have already seen like how technology is helping us on diagnosis, on having a continuous diagnosis on how your heart is pumping, how we can control glucose on your blood. We can actually monitoring continuously different molecules in the human body. Nanotechnology is working towards design very, very, very sensitive sensors, very small. And the philosophy behind that is that's very specific, so they can actually detect very few molecules of a disease. For instance, very few molecules of the presence of a bacteria, for instance, at the point that you don't even have the symptoms yet. So we could detect that you may have you can have a disease before it happens. So this could be really useful when hearts start on other devices, on other diseases. And on this picture here, that is kind of a picture that where we are working on having something very small that can diagnose a disease even before you have the symptoms. On nano devices and medical surgery, this is pretty cool field of research. Basically, you have a picture there of the first prototypes of macro robots, and you can see here a doctor playing with it, actually making the chirurgy onto the human body, which is actually a learning. This is a plastic body, and how they can control with better, precise movements. So nanotechnology is designing. Imagine that you can change. So you can see here the body. I don't know if you appreciate that detail, and you can see here all the tubes and things that are going on inside the human body. So it's still very aggressive. So imagine that we can change to this. This is a nanoneedle. This nanoneedle has a diameter of 60 nanometers, even smaller than that. A nanoneedle that can actually go through your skin. It could go to a cell for cell therapy. It could go in between the cells. It could actually remove one cell, and there's a hole inside, for instance, like a shrink. So you can actually drop drug inside the cell, or you can remove the cell. You can actually do at a very precise level. One thing more important on this is that you will end up not with any scarf. It's very little aggressive for the human body. And this is more or less what on the nano devices for medical surgery is moving on right now. On nanotechnology in regenerative medicine, where we already have a very nice talk on the 3D printing, in nanomedicine, we're going even further on that. We want to have, imagine that on the 3D printing, we already have some organs made on plastic, on different polymers. But the idea in the future is to have 3D printing that the bricks, as you were talking us, Eric, will be cells. Imagine that we can have cell by cell in a plane to form a tissue, like artificial, like the first prototypes had been done, the artificial skins. We need a lot of research to get to this point because we still don't know quite a lot of the biology of the cell. So these first prototypes are starting to work with stem cells. Stem cells are very promising, but with little we know now. Basically, the problem that you have with them is that when you start growing and they produce the differentiation, they actually don't follow the path that you want. And that makes sense because the cells are not just around your body, they actually are on what we call extracellular matrix. Okay, so in nanotechnology, what we're trying to do is mimic that extracellular matrix, as you can see here, one made with nano wires, put on top the cells, and that we want to make these nanowires like a nanoscaffold to indicate to the cells where they should go, and the most important thing, when they have to stop growing. A lot of the preliminary experiments that my friends are working on this, they always tell us that, yeah, they follow, but they never finish on working, so they're producing really bad consequences. So this is one of the applications on regenerative medicine. Obviously, at the same time, what we would like to have is to obtain these nano networks with nanowires, and that you can actually add growth factors, not only cells, but with growth factors or different drugs that you can be injected on the spot to activate and recover a damaged tissue. My favorite one, nano drug delivery systems, which is probably one of that we have, as Eric has explained, a lot of different ways of doing a drug delivery system. The philosophy behind this is like, as you know, when you take a medicine and you took it like a pill or in any other way that it goes inside the human body, the medicines are very, very unstable. So they degrade it, and basically nearly 3% of the medicine that you take actually reach the pain. So the philosophy on the nano drug delivery systems is actually increase that percentage. So how we do it? We design nanostructures that we can confine the drug there. So just by that, we are protecting the drugs. And we're going further. We want to target to the place where you have the disease. Obviously, because I'm here, I'm very happy that instead of talking about other people's work, I will present what I'm doing on this. And during my PhD, my first postdoc, I was actually working on discovering new chemotherapy drugs, new drugs against tumor cells. And one of the things that I've been thinking for all this time is that we already have plenty of drugs, okay? But the problem is that they don't reach the tumor, or they're very unstable and they are degraded. And more than that, the chemotherapy drugs that you actually take not only goes to cancer cells, but they do go to all the other cells that are still alive in the human body. So as a consequence, we all have all these secondary effects of chemotherapy. So I got tired. It's like, okay, let's find a solution. So how I can do it? So I heard about nanotechnology, about these new properties of material at the nano level. So I started looking at the literature, so what I could do. I chose several of them because I didn't have, to be honest, quite a big spectrum of what will work. That was in 2010. So it was very early on the properties of the material. So I didn't know what material could work the best. So I learned how to work with materials, and I was targeting into deliver the drug into the cancer cell. As soon as I started, I have a problem. Everything that I was following, I was doing the protocols I was following on the paper, everything I was doing perfectly, but when I have this nanostructure made and I put it into liquid, I ended up into problem of aggregation. So that's another problem that we have when working on the nano level, because we still don't know, we still at that point didn't know why this is happening. So a lot of very basic research needed to be done before I can actually think that that could be something similar to a medicine. You don't want this to happen in the human body. However, what was my problem was my inspiration as well, and I decided to find a solution for this problem. As Eric talked about the DNA, one of the most beautiful, from my point of view, molecules that we have in the human body, I decided to play around a little bit, in vitro, as they say, with the structure of a double helix. It's a circular double helix, so I can over-twist or under-twist the DNA. So I thought in a very easy way. So I thought, like, okay, let's do, so that's a cartoon. This is a real picture I see in the lab of the DNA. So I thought to do this reaction in the presence of carbon nanotubes in order to have the DNA wrap along the surface on the carbon nanotube. On the bottom, you can see a cartoon of the carbon nanotubes. Close to it, the carbon nanotubes all aggregated. After 5 minutes, I made them. It's like a huge aggregation. And then after several trials and a lot of thinking, I managed to have, as I wanted, all the surface of the tube wrap the DNA on the surface. So in that way, I managed to have, so you can compare it from them to here, so beautiful carbon nanotubes that are soluble, stable. So I check the stability in different liquids, not only in water, because if I work in medicine, you have to choose proper physiological mediums. So I choose the ones that you work in vitro. I actually check again as well for stability of temperature and pH, very important in medicine. So it's actually pretty stable. It allows you to work in quite a lot of range of temperature. And one of the most important things is that it's reversible. So what I mean, when you let's put it together, two things in a self-assembling kind of way, and you don't have to use any extra energy to produce this change, you can actually make it reversible. Why is this important? On the drug delivery systems, so what you want to achieve is you have a nanostructure, and you can put your drug. You need to close it because it needs to be confined. Go to the human, go to the body, find the place, go inside, in my case, on the cancer cell, and when it's there, open. So this is the reversibility I'm talking. So I managed to prove that I can do it in vitro, and I thought, okay, if I'm right, the same thing I observed in vitro, because I studied properly the conditions, are the conditions that these nanostructures will encounter as soon as they go inside a cancer cell. The inside cancer cell was the easy part. Cancer cells are very permeable, so it's very easy to absorb, that a cancer cell absorbs these nanostructures. So to prove this is a preliminary experiment where you can actually see that as a function of the size and on time, you can see from the starting point in 25 hours, I reduce the viability of cancer cells from 100% to 5 to 10%, so I'm trying to get it better. This is only the beginning, and this is to prove that the system is working. Obviously, these experiments are done in the lab with cells, with cancer cells. You add it, everything is perfect. That doesn't mean that's going to work on a human body, so I'm moving towards like a complex organism now. But at least this reversibility is working. So this is to summarize what I'm doing. So basically, the first part was obtain the nanostructure, and you make a little bit of porous on the surface so that the drugs can be absorbed on the surface. The second phase is what I have shown to you now. You have the carbon nanotube, two different drugs, or one drug, chemotherapy drugs, wrapped with the DNA. And I proved and I showed to you that this can go inside the cancer cell. However, I'm going to go further. So I would like to have a way that these nanostructures can actually target the cancer cell. So in the lab, it's very easy because you only have cancer cells and control cells, but in a human body, you have a very complex system. So what I actually have done in the last year is to be able to chemistry, link on the surface on the DNA. It's a little link, and on top, this blue square, I add one of the molecules that is overexpressed and is presented on the surface of a cancer cell. So the phase number 3 is targeted through molecular recognition of cancer cells. The good thing is that the link, that reaction is done in 2 steps. So basically, you can add one protein, you can add a fragment of an antibody, you can add whatever you want to target molecular recognition on the surface of the cancer cell. Now, phase 4, I'm still there. this is my way of where I'm going now. So I want to work on diagnosis, basically because this is quite universal, so I can actually add a fluorescent marker. I can actually add radioactive, so we can use them for target and visualize where is the tumor. And obviously, in the future, we want to work on diagnostic, which means diagnosis and therapy on the spot. So this is what I'm actually working on. So this is my nanosystem, what I call it. Basically, what we have achieved is that the structure is stable, and actually the drug is stable. You have the reversibility, so it means that it can open and close, and you don't need to irradiate the nanostructure to anything. It's only because inside the cells, the DNA will unwrap, liberating the drugs. We are working on making selective to cancer cells. And number 4 is universal. So what I mean, what is universal? And this is pretty cool, to be honest. So what I thought is like, okay, I have this way of wrapping the DNA on a carbon nanotube, but I thought that the way that I'm doing it is not just only in carbon nanotubes. So I realized that I can do the wrapping on different type of nanoparticles. And at the same time, in parallel, we were working on synthesis of silver nanoparticles, and we realized that they are very toxic in bacteria and microbes. So toxic, let's say that they actually toxic on bacteria, so they have a high activity of antimicrobians, and basically is related to the instability of silver surface nanoparticles. However, if you think about it, if you can actually confine the surface until the moment that you want and target to a specific bacteria, okay? And this is what this universal system can actually do, becoming very useful to study new potential antibiotics. And the idea of targeting is that, because like in the cancer cells, you have molecular recognition, but when you talk about in the human body, you know that you have quite a lot of bacteria in your human body that you need it. So you don't want to target all the bacteria. So we're working on the same way of thinking, on the same philosophy to targeted bacteria as a way of a new antibiotic tool. Overall, what I say, I would think that the major areas of development of nanomedicine are, as you can see from all the different blocks that I have presented, is to have a better prevention and control, especially with the nanodiagnosis, early detection of a disease, better imaging diagnostics, and multifunctional therapeutics. But I have to be fair. Nanotechnology is helping a lot in medicine, but medicine is helping a lot in nanotechnology. It's something that a lot of nanotechnology applications, they don't care, but it's about nanotoxicity. As medicine, we work with bacteria, with cells, with viruses, with human body, with organs. We know we can inform to the world of nanotechnology, this material may have these properties that are pretty cool, that they do what you want, but this is very toxic. Or inform to nanotechnology applications that has nothing to do, obviously, in medicine, but be careful during the process of synthesis. This is quite toxic, so please, when you do the synthesis of the nanostructure, cover, make yourself quite as safe as you can, or as well inform to nanotechnology applications that has nothing to do with the medicine or the human health, as they say, but for instance, when we use it in our smartphones or in our computers, you don't think, okay, if it's toxic, I don't care because it's in my computer. I mean, I don't eat it. There's no problem with that. But you know that every 2 years we're changing computers, we're changing telephones. So you are creating a nano waste at the end. So medicine is helping to determine what is nanotoxicity, what makes things toxic and what makes things not toxic, and inform to the world of nanotechnology and other applications about this. So it's a kind of nice relationship. Now, every time I gave a talk, they always ask me, okay, Sonia, when we will see these medicines in the market? And this is the worst question I always have. I don't know. We still, in my point of view, this is my opinion, we're still in the innovation phase. We know more and more and more. We know how to make things. We know probably 10% more or less of the properties of the new materials. We need to know now the properties of hybrid materials. When we finish the phase of innovation, obviously will be development and actually finally get what we call nanomedicine. And how long it will take is actually, it depends on the decisions that we make now. So at the Oxford Martin School, what we like a lot is to think what's going to happen in the future and how the decisions that we make now will make that this go faster, this go slowly, or one application go first and stuff, and things like this. I don't like to talk about problems, but let's call it challenges. We are facing a lot of challenges. For instance, we're having less and less money for basic research, which is a mistake. We need to work very close to make a proper regulation, and we actually need to quite a lot of time. Okay, but basically one of the biggest problems, at least from my point of view, is funding projects and especially funding people. So just to finish, to say that basically the end goal of nanomedicine is basically improve diagnostic, treatment, and prevention, but the ultimate goal is to integrate diagnostic, treatment, prevention of one disease into a personalized healthcare. What I mean is that the more that we know about our bodies, the more that we know how we will react, the better medicine we will have. Like for instance, when you hear about a person having a tumor, so we know that in two different persons have with the same type of tumor are completely different. Some people will actually, but all of them are treated with the same type of drugs, and so we know that some of people will not react properly to the drug, to the other one. So the more information that we have using the different tools I showed to you today, to know more about a person's body and how they will react on one typical medicine, the more benefits we will have, and actually we will obtain the better healthcare. So thank you very much. And now we open the questions and answers.
Well, thank you so much to Eric and Sonia for that. It was a whistle-stop tour, but the great thing is that you will be able to re-watch it online if you feel that that was a lot of information to absorb very quickly. Does anybody have a starting question to kick us off? No, well, the gentleman in the red jumper.
I wonder if you find it surprising that today most of the nanotechnology is done with actually rather large and elaborate machines rather than self-assembly. And does it matter? After all, we can do things, so does it matter that it's not self-assembling or
there are many very different technologies that share nanoscale structure. And all of those are called nanotechnology, but they're extraordinarily different. As Steve Jurvetson of Draper Fisher Jurvetson said some time ago, do we talk about centitechnology, millitechnology? We don't ordinarily class technologies by size. And I would just mention that we have an overwhelming nanotechnology-driven revolution today. It's being driven by nanoelectronics, by which I mean microelectronics, which is now deep in the nanometer size range, tens of nanometers for transistors. So that's an example of large, expensive machines being used to make some very intricate and very useful objects. But self-assembly and atomic precision is another frontier.
Thank you for two fascinating talks. Dr. Drexler, you mentioned work with the DOE in the States. I wonder if you or Sonia have any comments on military applications of nanotechnology and where that's going and the anticipated threats and benefits of that kind of research, more DOD than DOE.
Any comments on it? There's some defensive applications in the nanomedicine area.
You first, Eric?
Yeah. Well, the answer, of course, is yes. The DoD is interested in, and general defense departments everywhere are interested in every technology that expands human capabilities in just about any area. There's a lot of medical research that's driven by military interest, space, electronics, materials, organizational development and psychology, pretty much all areas. The prospect downstream as this atomically precise manufacturing technology matures, climbing a ladder of technologies to something well beyond what I described here, like the difference between a blacksmith and an automated factory, will change physical technologies in a foundational way. higher performance and lower cost, and that is among the many different future developments that are going to be revolutionizing the military world. There's large arms control and strategic stability dimension to these technologies.
Yeah, and actually, not only in that part, but there are lots of interest as well to develop something different, but new nanotechnology textiles. Okay? So thinking about not only to enhance the human capability, but actually to have a better suits where like they could keep warm, not wet, like so nanomaterials that actually you can have like proper shoes, proper shoot, everything. So to keep them warm or more protected. But on that, yeah, this actually nanotechnology in textile is working on that.
Right at the back.
I'm curious about the drug delivery nanosystem. I might have missed some information from your talk, but how would the body metabolize and excrete the DNA wrapped around the carbon nanotubes with drugs on it? And
so, yeah, thank you for the question. So the size is important. The size that more or less I'm using right now is between 50 and 100 nanometers, but 50 is one of the best. At this size, the complex that is not being used by the body will be going out through the normal kidney urine system, so there will be no problem. The system that will go inside the cell, the DNA unwraps due to this reversibility property. The DNA at the cytoplasmatic level of the cell will last nanoseconds. It's been degraded. Left a nanotube of carbon and the drugs. The drugs will do their own. And the carbon nanotubes, it's really, really small, okay? And are actually, at the ends, are chemically modified in order to produce the biodegradation of the tubes, leaving into very small pieces of atomic carbon, which will be definitely not bad for the cell, but actually you want to kill that cell. So, but this is more or less how the system is done.
Yeah, I would note that with a greater flexibility of fabrication, more choice of building blocks and sizes and so on, biodegradation and metabolic considerations, you could satisfy those constraints while also satisfying the medical delivery constraints.
Who's next? Up at the front here.
Thank you so much, both of you, for an extraordinary illuminating talk. Can I ask the same question that you were asked earlier about warfare, but this time about food and, as it were, feeding the world, and the impact of nanotechnology on that?
Well, there are a wide range of nanotechnologies again. What I can speak to most directly are the prospects associated with advanced high-throughput atomically precise manufacturing. There, the main first-order effect is going to be lower cost of capital goods, more efficient, low-cost way of transforming common materials, carbon, silica, and so on, into high-performance hardware. Well, why don't we have enclosed greenhouse agriculture today widely used? It's because the structures are expensive. Having manufacturing the structures needed to build greenhouses and to make them easily deployable and put in place, it's a substantial cost. If those were much less expensive, you would be in a position to expand that kind of agriculture, which turns out to increase agricultural productivity by something like a factor of 10 per unit area. It's really quite striking, and that's for grain production where nobody uses greenhouse agriculture, but experimentally that's the result. And if you had that, you would be able to take the footprint, the ecological footprint of farming, roll it back, and have a wide range of other advantages, such as exclusion of pests, better recycling of nutrients, and so on. So lowering capital costs, improving manufacturing generically can affect not only much better military hardware, but at a lower, driving the cost lower, agriculture.
So actually, let me point out in some of the sensors that I was talking, they are being used in nanofood as well, so they can actually detect CO2, for instance. As in, let's imagine that you have a plastic or food, like wrap, and they have these nanochips there, so they can actually detect molecules like CO2, which will relate it to the presence of bacteria already on the food, okay? So sensors to see how the quality of the food is. And as well, there is a lot of prototypes on producing a plastic made with nanostructures, a plastic that is biodegradable, but not in 20 years. Biodegradable after you use it. Okay, so if you think how much plastic you have at home, every time that you buy some cheese or some ham, you end up having more plastic than actually cheese. So one of the applications is actually to change this type of plastic to a plastic that will degrade in minutes, okay, not in long time. And as well as he mentioned it, there is a lot of nano on agriculture. So nanosensors for detecting where is the infection, so where you have to drop pesticides. So instead of actually putting in all the land, you know, like you know where it is, so you just go there and target there. So millions and a lot of quite of applications in food.
So go to Julian next and then go back.
Yeah, thanks very much. This might be an outdated question, but is there still a concern about runaway self-assembly and consumption of organic matter? And what are the controls on self-assembly?
Well, let's see.
Around?
The self-replication is the concern.
The
Yes,
grey goo sort of-
yes. I used the term grey goo about 4 times in 3 paragraphs in a book in 1986. In the context of saying, well, in the range of machines that could build more machines, if you took the trouble to make them operate in a very hostile environment, which is to say nature rather than a laboratory, and you designed them and programmed them to replicate indefinitely and some other conditions, then in principle that would be something very bad. You don't want to do that. Instead we do these other things. Then there was an article in something called Omni magazine, which reached 1 million people at the same time my book came out, which had these couple of paragraphs. And the one pullout that had large print was grey goo. The cover was nanotechnology, the first time most people had seen the word nanotechnology, which I introduced in the contemporaneous book. Molecular machines that mimic life, and ever since then, it's about little things that are like life, little nanobugs that might eat you. And that had a shred of basis in the formulation that I had in 1986, a big, thick technical book titled Nanosystems, 1992. Said, well, you really don't want to build anything even remotely like that for practical reasons. Factories should be sort of large if you want to make large things, and most of what you want to make is large things or substantial quantities. So the answer is, worry about arms control, not someone making pointless nanobugs, which is only exciting because science fiction authors in the press ran with it after the Omni magazine article in 1986. It's been a zombie idea through the world.
Then there's 2 questions.
One of the major challenges that medical science and humanity face at the moment is to be able to understand the behaviour of the proteins that are badly folded in Alzheimer's disease because they are intracellular. When I watched your nanotube with the DNA helix wrapped around it, being inserted into cells, do you think there's any future in putting proteases around nanotubes and inserting them into the hippocampus to try to dissolve these plaques?
Yes, actually I have good news for you. I will not be the one doing it, but it's very close. I know that it's going to be very soon. If it goes to the final clinical trials, there is a medicine that works with gold nanoparticles. Gold nanoparticles do interfere with the amyloid plaques. And when irradiated in a very near to infrared, so not really bad light, I would say, they use these properties of the nano level would actually degrade the amyloid plaques. So I know that there is nanomedicine going on in this way, and I hope that see the light, but probably that's one of the first nanomedicines that we see on the market. And the way that these nanoparticles have achieved this when you think about it, you can actually use absolutely this type of thinking to target the place. So the problem with the Alzheimer is that the amyloid plaques is not why you have the Alzheimer, it's a consequence. So we still, at least we will win some time to know why the disease is happening. So the amyloid has the problem of dementia, but it's not the source of the disease. So we need to look why we have these problems on the Alzheimer. But yes, there are good news on that.
The next question was, do you still have a question? Yeah.
Yes, it's related to Professor Savulescu's question, but perhaps a little less dramatic. I mean, if we discard grey goo scenarios, what are the regulatory and governance challenges that we should be thinking about now in relation to nanotechnologies?
The ones that I find most striking are involved in downstream technologies, well beyond what either of us are working on or looking toward in the next generation of technology, where the prospect is for a transition somewhat like the Industrial Revolution in scope. You know, the controlled programmable fabrication of things, but it turns out lower cost and higher performance are in the cards, with implications for the economy and strategic balance and arms control. My greatest fear is that as these technologies progress, military planners will say, here is a way of making better weapons for fighting our wars. And the wars they will be planning for are wars that will no longer be motivated in an economic transformation of that sort. If you look at the drivers of conflict, other than just the spirals that run in the South China Sea and many other locations, it has to do with trade and raw materials, both of which are going to be radically transformed in their role in the world by a revolution of this sort when it comes downstream. And I think there will be a requirement to rethink national interest in a fairly basic way in connection with these technologies and the military dimension. So it's a bit beyond regulatory framework and it's downstream, but it's what looms largest in my concerns.
At this moment, we have regulation nanotechnology in 3 different applications, which is food, cosmetic, and medical devices. Obviously, everything that you do in medicine, you have to go through clinical trials. So if you ended up at the end of marketing, that has been properly regulated. But I agree, there are some parts of nanotechnology still not regulated, and what is one of the things that actually our roles as well is to be informed to the regulational bodies what's going on and transmit our knowledge, because it's very difficult to regulate something that we still don't know. Okay, so it's our part as well of working to inform them and work together.
Yeah, I would note that the spectrum of nanotechnologies that we're most about today in nanomedicine, nanomaterials, and similar areas, the concerns are conventional from a regulatory point of view. They have to do with toxicology, medical trials, releases into the environment. There are novel scientific questions because nanomaterials aren't the same as chemical compounds. But stepping back a little bit, it's the same kind of question.
Just behind you then. Yeah.
Thank you so much for your lecture. In plant systems, we have a problem, as you know, of these nutrient use efficiencies. And are you able to hear?
No, really, I cannot hear you properly.
If you hold it a bit closer.
In plant systems,
Yes.
We have this issue of nutrient use efficiency problems because of which these fertilizers are being spilled. And do you see any possibility of developing something like nanofertilizers? And is there something of that future in the next 20 years, something?
Yeah, I mean, the different, when we call about this nano-drug delivery system, change the drug to vitamins, any kind of other molecule. So all these drug or all these delivery systems, in my case, for instance, I work with drugs, but if in case of delivery, for instance, vitamins, nitrogen, whatever the plants they need. So it's the same way of thinking on targeting the plants, the deficiency of nutrients. I mean, for sure that it's another, so you have to think of the different delivery for nanopesticides. I'm sure that there will be and I'm sure they will need to be regulated, and probably with high efficiency. And then you will have to face again organic, inorganic, and nano pesticides. So it's going to be a great debate. I hope to be around.
Who's next? Sorry, Clara, up here.
Thank you. A question for you, Sonia. I would like to ask your opinion on the toxicological study of nanomaterials. So, for example, gold or silver nanomaterials. Do we know enough? Is there enough research done on the toxic effects on human body to go and use them in medicine?
Thank you for this question. I think that in the last 3 years we have done a lot on this research, especially in gold and silver, because are the nanoparticles that have been more used in different products. And particularly, so you asked for my opinion, I will tell you what happens on my research. I started working with silver nanoparticles, and with my student we realized that it was really, really toxic with bacteria. So at the point that when we finally did the proper assay of toxicity, and that was the clue, when we saw it, it's like we step up, like wait a second, it's a very silly reaction because we were working with them for like about a year or something. So what we find out is that the test of toxicology that it was going on, it wasn't really good, it wasn't accurate. Okay, so we have new materials, new properties, so you have to tailor the protocols in order to check the test of these new materials. Say that we have in my group developed, I hope that it's going to take us a standard protocol for nanotoxicity, because one of the things that I realized, especially with silver, when I went to one conference, there were 42 talks saying silver is toxic in fish, silver is toxic for plants, silver is toxic for everything. I'm like, wow. But the problem is that they use different technologies, so we couldn't compare the results. So I thought, okay, what we can do, let's work and make a standard protocol, a protocol that is simple, but very reliable and robust, so everyone can use it, from physics, engineering, bio, everyone. So you have an idea on that. So also, we went through and we are in the process of generating a database for all the different nanomaterials, because the properties change as a function of size, shapes, material, so it's complicated. But for you, I tell you that silver is toxic. We know now why. Unless you confine the surface, silver is toxic. Okay, it's the release of the ions from the surface will make that toxic. But it's interesting. Gold is perfect. No problem of toxicity with gold. Spherical nanowires, we found no toxicity when gold is nanostructurate. Okay? But yes, that's what we're working on.
Any more struggling to see? I think I have one question, which is about the scale that research is happening at. Obviously, you flagged the perennial issue of funding for novel and high-risk research. Do you have a sense of globally how many people are now prioritising research into nano in the way that you are? I mean, particularly Sonia on the sort of practical side, but also obviously you are.
Well, if I see the number of papers that you have published on nanotechnologies, that is scary. We have 4 papers per day for articles. It's impossible to be totally updated. Obviously, not all of them are scientifically relevant, but we have a lot of, so that could be a measure of, there's a lot of things going on, especially the drug delivery system, I feel, is quite competitive. But, okay, let's hope to have a perfect drug delivery system. Now, the problem is that now we are in a situation that it could go in a A or B pathway. A will be exponentially going, B is like slowly and going down. So it's interesting to find funding for these projects, but one of the things that I mentioned is not only the projects, you can have many projects, but if you don't find the money or the way of contracting and having the researchers working in permanent positions, so all these ideas will go away. And this is where we're having problems. Funding on PhDs, postdocs, permanent positions for researchers, and if we don't resolve this issue, which is really important, all these new ideas, all these new things that we are doing, they're going to develop for sure. Technology always goes forwards, but it's the speed and the quality that will be actually affected.
And is part of the issue how to engage the pharma industry to to give this support? Is that one of the issues?
Well, in my case, working on pharmacology, and when I talk with pharma companies, they have it very clear. All the companies are really interested in nano, but for them, obviously, it's business. It's too risky.
Yeah.
Okay, so they're always extremely interested. But what I got is like, okay, when you pass phase 1 on the clinical trials, we will be involved. For me to go into phase 1 clinical trials, it will be now like £5 million. I don't have them. Okay, not at all. So there is a gap between research, basic research, applied research in the lab, and then the clinical phases. So there is a gap here. But we are working on new models of funding and see if we can find something that actually help us to work together. Because nanotechnology, it's like an industrial revolution, but something has changed. And it's the way that we do science. We actually work together, people from different disciplines, medicine, biology, physics, math. So now it's time to bring the companies to work together with us.
Anyone else? Yeah, the gentleman in the check shirt.
So I'm particularly interested in the applications of nanotechnology on basic research as an aid for basic research. So in particular, biology. There is this problem that if you want to study the biology, especially like the intact organism, the probes that you use to study that biology basically cause a massive damage most of the time, or they modify the system in a way. So it seems like there could be a lot of potential to develop probes for basic biological research using nanotechnology, but I just wonder if, like, what are the prospects? Is there something that people are thinking about or working on?
Theranostics?
Yeah, well, I have to tell you, I'm a biologist. Okay, though I work in the physics department, I'm more like basic research on biology. So I found very useful nanotechnology for studying biology. Not only the nanoparticles and the nanostructure, but all about the new microscopy that allowed us to observe things. So why I moved from biology, basic research to physics, which is a little bit weird, is because I wanted to learn how to work with atomic force microscopy, which is the only microscopy that we have right now that I can visualize things at the nanometer resolution in liquid. So if you're a biologist, you don't want things in air. You want things in liquid, okay? And so I learned, it took me quite a long time. We did high-speed atomic force microscopy, so we were able to see the first molecules like thermal fluctuation, which means moving in liquid. And definitely for a biologist, when you see, you understand a lot. And because I start seeing the DNA, the isoproteins, you can understand problems of how they fold. You see cells, very, very precise details of the membrane of bacteria, and that's the reason that I'm still in the physics department, actually. But yes, it's advancing a lot of the knowledge on biology and basic research.
Yeah. So, very interesting example of the kinds of systems people consider when they start thinking about complex macromolecular architectures. MIT and Harvard, there is systematic exploration of the feasibility of having nanoscale devices, instruments, that could, in the brain, record patterns of electric potential, neural spiking, and so on, in what amounts to a ticker tape, DNA, encoding in a DNA strand what a sequence of actions was, time-resolved action in the neural system, and then have that information be taken out in the form of that strand and read. Now, that's being considered as a serious prospect. I think not likely to work, but the point is it's in the range of being worth considering with self-assembled systems. If you're able to build systems block by block, try it, find it doesn't work, do some debugging, build another one, build 10,000 variations and try all of them and see which ones work, and all of this at the level of large, intricate structures, you can probably make that kind of scheme work, and many others besides.
I saw another hand. Up at the front.
If the barriers to development that are there at the were to go away, what is the kind of timescale that you think that you could get to the kind of manufacturing that you're talking about in?
Well, what I described here was what I think of as the first rung on this ladder of mechanically directed assembly, where we've climbed a long way based on self-assembly, making that transition. I've discussed this with a number of people who've, in groups that have experience in development and integrating systems. And I know that at the DOE workshop, the time horizon that was being discussed was 5 years with, if one mobilized multiple groups to work on each problem so that you had confidence that at least one solution would be put forward for each of the component problems that would be integrated, where you take those and integrate them to make the system. And I think that that time frame is realistic given the right kind of project management and the right funding and marshalling the kind of enthusiasm that I think is already there, but not presently organized.
In terms of it getting to perhaps the medical application stage?
That's where the delays, that unpredictable and large delays come in.
Well, I mean, you have to go through, so imagine that on the nanotubes and the cancer cell, okay, you have the preclinical in vitro, which is what I'm doing, so the cells in the lab, you put in, incubated, then you go to preclinical in vivo, is when you start working with mice, rats, then you go to phase 1, when you have proved the efficiency of the drug, then phase 2, phase 3, and then market regulation and market. 20 years, more or less. Interestingly enough, this year we will have a liposome with cisplatin medicine, okay, that the medicine by itself we've been using it quite a lot right now. The time that I heard about this was in 1995. So let's make medicine for our grandsons. But that's the scale, sorry. I cannot go fast.
Perhaps have faster fabrication, experimentation, testing, and move the development and approval pipeline to Kazakhstan.
But they were at -50 degrees today.
Where recently in Astana it was -50 centigrade, which is...
Sorry.
Yes.
Yeah.
I'm not moving there.
No, neither do I. No, but actually it's true that one thing that we can do is to be more organized. Detect which groups or which places are they doing this, and this, create a platform to where we can identify the main research people and actually collaborate. Because, for instance, in the case of nanotoxicity, I realized that a lot of people are doing things that they will never see the light. So, hey, be careful, change it, and it was changed. And silver, products that were already on the market with silver, they were out. Okay, so I think that what Eric's saying is actually true. We need to be better organized, and it's not easy to do that. But if we manage that, I think that the speed of the development will be exponential.
And one final question back to the gentleman here.
It's a great responsibility. Thank you. You've been talking about things and processes, very small things and very small processes. In one moment there, you talked about the nano getting into the brain and starting to take records there. Can you imagine this enormous change that you were talking about in productivity and diagnosis, healing and so forth, also having cultural consequences? Might one say nanoculture, nano-information, nano-spiritual, and mean anything?
Well, the nanotechnology revolution today, again, is nanoelectronics, which is in our cell phones and computers and is radically transforming the world in, I think, pretty much all of the ways that you mentioned. Transpose that into the world of manufacturing and medicine, and I think that the prospects are beyond anything that we can hope to understand or predict. I hope that some of the principles of institutions and human values can be applied to guide these processes to some destination that we think is good. There's obviously the possibility to do a lot of good with new capabilities, and also the possibility to inadvertently cause disruptions that are extraordinarily adverse. Now, some people say, oh, Eric, you're talking about these great things we can do in the future. You must be an optimist or a utopian. My view is no. I think there's a tremendous potential upside, a tremendous potential downside, and change of the magnitude and speed that seems to be possible is one that I find scary. Now, I would note that my day job today is AI safety. And that's an area which is also moving extraordinarily rapidly. Anyone who is interested in where the world is going, read about deep learning, because now people are doing AI, I'll put this slightly radically. The great thrust in AI today that is moving rapidly and beyond what historical precedent would have suggested is, in a sense, not based on software. There are relatively simple mathematical structures, a lot of matrix multiplication, differentiable functions, gradient descent in high dimensional spaces, not if then, count to 5, move pointer, all those things that software does and still happens underneath the layer that I'm describing, but the layer at which adaptation occurs, at which learning occurs, at which algorithms are executed, is a layer where people do not know what the representations are, they did not code them. They do not know what the algorithms are, they did not code them. They're learned, and it's moving very fast. If you were paying attention 2 years ago, you have no idea what's going on now.
Before we thank Sonia and Eric, I'd obviously just like to highlight a few events that are coming up. Obviously, the seminar series continues at this time every Thursday. Next week, it's Hannah Maslin and Roy Cohen-Kadosh on cognitive enhancement devices. But if you aren't aware of the whole series, there's a leaflet outside or obviously our website. And just to flag that our International Women's Day lecture this year, which is on the 2nd of March, will be Judy Weichmann from the Internet Institute, and she's going to be looking at automation, robotics, and the promise of an easier life, which for those of you who are interested in technology's impact on humans, I think you'll find that a fascinating lecture. Plenty more besides, as you know, you regulars will know that we have a whole heap of events, so please do look at the website and sign up to the email to make sure you're the first to hear about our events. But I'd like to thank Sonia and Eric for a truly thought-provoking seminar. I think plenty of optimism, plenty to think about, and let's just hope that the powers that be will marshal the correct resources deployed in the right way to make particularly, the nanomedicine benefits happen sooner rather than later. Thank you.